All right, so microscopes. Microscopes first started to be sort of invented in the 1600s, and it wasn't until really glassworking started to become sort of well-known in ways of grinding lenses and forming lenses. That allowed microscopes and telescopes and things like that to start to be developed. So as lenses got better, microscopes improved, scientists were able to see things at a smaller and smaller scale. And so some of the first microscopes were really simple. They don't look like much. That's one of Leeuwenhoek's first microscopes. And so the way this thing worked, you would hold it up to the light. You would be looking from the opposite side that's not shown. The lens is actually embedded here in the brass. You would put the sample you're looking at the tip of this sort of needle here. And you could adjust the focus by turning this knob and moving it farther or closer to the lens. You could adjust, where it sits there. So that's kind of what some of the first microscopes looked like. Leeuwenhoek he made these types of microscopes. Sometimes he's called the father of microbiology. He came up with sort of a unique way of making really high quality lenses, but he was very private about it. He didn't want anyone else to know how he did it, so he didn't share his lenses with other people. He didn't tell people how he was making them. People would come over and want to use his microscopes. He would allow them to use like his inferior ones, but he saved his best microscopes so that only he could use them. And he discovered lots of things like bacteria, protists, cell vacuoles, muscle fiber. He saw all of these things using these really simple microscopes. Turned out later on they found there's I think 9 of his microscopes still around today. They found that what they think he was doing was melting glass, dropping little drops of glass into water, where we then solidify and make these very perfect tiny little spheres, which gave a really high level of magnification. His microscopes could magnify up to 275 times, which is a little less than what we do in our microscopes here, our modern microscopes, all using this sort of simple technique. Robert Hooke was another famous scientist who was early in examining cells. He published a book called Micrographia in 1665, and basically he had all these drawings of things he observed using his microscope. He's the one that came up with the term cell. This is what it looked like, the micrographia, the front of it. Okay, looking at these tiny little things, and he made all these drawings. This is cork, and this is where he came up with the term cell. Okay, he looked at these little compartments that were in the cork, and he called them cells because they looked like the cells that monks slept in a monastery. So he drew all sorts of things. And once microscopes were invented and used commonly, that allowed people to see things that are microscopic, obviously, cells and blood and bacteria and single-celled organisms that otherwise nobody really knew existed before microscopes were invented. This is one of Hooke's microscopes. Looks a little more like our traditional microscope. You might think of it had an eyepiece, had objective lens. They used candlelight or sunlight as a light source, because obviously there was no electricity at the time. So we have 2 microscopes that we use and scientists use all the time. The compound microscope is the one you're probably most familiar with, that we use a lot in 7th grade. The compound microscope uses light moving through the specimen to create an image. So whatever you're looking at has to be quite thin. It has 2 sets of lenses that magnify the image. There's the eyepiece lens that you look in, and then there is the objective lens. And most microscopes have several objective lenses that you can select. To calculate the magnification power in a compound microscope, you multiply the eyepiece power times the objective lens power. Our microscopes that we're going to use go up to 400 times. You give a 40x high power objective and a 10x eyepiece, but there are light microscopes that could go up over 1,000. Often those are called oil immersion lenses. We have a couple microscopes that have one of these lenses. Basically, it's a 100x objective or higher, but in order to use it, you have to put a drop of oil on your slide, And when you select this 100x objective, it also touches the oil. So rather than the light traveling through air, it travels through this oil and allows it to have this higher level of magnification. You might use those in college. When you look at a compound microscope, what you see is inverted, upside down, and it's also left to right, backwards, due to the lens. So there would be an image from a compound microscope. What you see is flat, very thin things. Our microscopes have a pointer in them that if you rotate the eyepiece, this pointer moves around so that you could show somebody something. Put the pointer on a certain part of the field of view and point it out. The dissecting microscopes that we use, they have two eyepieces. And the difference between the dissecting microscope and the compound microscope are that the dissecting microscope uses reflected light. So the compound microscope used light traveling through the specimen. The dissecting microscope uses light bouncing off the specimen. So you can see three-dimensional objects. You could see larger objects, opaque objects, because the light's not having to travel through the specimen. Typically, these have two eyepieces. They're called binocular microscopes. And that allows you to get a three-dimensional view of the specimen that you're looking at. Typically, these go up to about 50 times magnification. Ours go 20 or 30 times, depending on which one you have. And again, they give you a three-dimensional view of whatever you're looking at. Joe, do you have a question?
Yeah, what's opaque?
That light doesn't travel through it. Sort of the opposite of transparent. All right, so how do we use the compound microscope? We use them lots of times, but it's always helpful to get a refresher because when we do a lab with the microscope, like we'll start tomorrow, the most common question I have is, I don't see anything. If you follow the steps, you should always be able to find your specimen. So we'll just review them. So you always want to be careful with the microscopes, carry them with both hands. So when people touch the objective lens or the eyepiece lens, it gets all smudged and fingerprints. So you can clean them with a special paper we have called lens paper. You always, always start on low power. That's one of the key things to know. You're going to start to view a specimen, you start on the lowest power that you have. You also want to put the diaphragm on the lowest possible setting. Diaphragm is that part underneath the stage that controls the amount of light going through. If it's overly bright, it makes it harder to focus. It makes it harder to differentiate parts of the slide. So you want to start on the lowest. But as you go to higher powers, they let in less light, so you might have to open it up as you are going through. We always focus under low power with the coarse adjustment knob, the larger knob. It moves the stage or the objective lenses more than the fine adjustment. So you look through the microscope, you turn the coarse adjustment until you start to see something. If you need to, you improve the focus with the fine adjustment until it's very clear. Usually in low power, it's pretty easy to find the object. It's easy to find it, easy to focus on. You want to center whatever it is you're looking at so you can see as much of it as possible. Our microscopes, you have to move this slide on the stage with your fingers. There are other types which I'll tell you about. Then, if you want to increase the magnification, your specimen is centered, it's very clear. Then you leave everything the same and you rotate to the medium power objective lens. When you switch to medium power, it should be there. You should see it. It should be pretty much in focus. If you need to make some adjustments, you could change it with the coarse adjustment, just moving it a little bit. You won't need a lot of adjustment when you go from low to medium power. Again, see your object, center it, adjust the diaphragm if it's too dim, and then you can switch to high power. Again, you made sure it was centered and in focus. When you switch to high power, it should be close. You should see the object still there. You might need to make some little adjustments using the fine adjustment knob only. You might need to adjust the diaphragm to help the specimen stand out a little bit better. Now, sometimes you lose it and you just can't find anything. If that happens, you just back down to medium or low power and start the process again. So whenever people are trying to find something high power and they call me over and say, I don't see anything, my first step is go back to low power, find the specimen, focus it, center it, switch to medium, focus, center, then switch to high power. Sometimes you'll make your own slides, sometimes we'll look at already prepared slides. If you're making your own, you can make what's called a wet mount, where you take an empty glass slide, you put a drop of water, you put your specimen in the water, and then you add a cover slip that keeps it all together. You want to put the cover slip on at an angle so that forces any air bubbles out, they don't interfere with your view. Sometimes you stain the specimen you're looking at. A stain is a special chemical that is absorbed by some parts of the specimen more than others. For example, iodine and methylene blue, they are absorbed by nuclear material. So if you add them to a slide that has a cell on it, the nucleus of the cell will become darker and more obvious. You may have to measure things under the microscope or calculate magnification. You definitely will. So again, total magnification is the power of the eyepiece times the power of the objective size. The units that we use when we measure in the microscope, typically we use micrometers. There's 1,000 micrometers in a millimeter. Typically our field of view on low power is about 4,500 micrometers. That's the area you can see when we have low power selected. Under medium power, okay, it's about a little less than 2,000 micrometers or so. And under high power, it's a little less than 1,000 micrometers. Here you see the parts of the microscope listed, and I'm sure you know those. Our microscopes use stage clips to hold the slide down. Just these two metal clips that keep it from sliding around much. There are some microscopes that have the mechanical stage, which is kind of nice. The mechanical stage has this bracket that holds the slide, and then to move the slide, you just turn these knobs underneath. One moves it forward and back, one moves it left and right, and it just allows you to be really precise in your movement. Lots of times higher end microscopes have this mechanical stage. We have a couple with that. As far as focus, our microscopes typically have two knobs. The coarse adjustment is the larger, and the fine adjustment is the smaller. But sometimes you'll find microscopes that have both of them built into one. The outside of the knob is the coarse adjustment, and then the inner part is the fine adjustment. It's all built into one control. No, I'm good. There are also electron microscopes. Electron microscopes, instead of using light to create an image, they use electrons. A beam of electrons is the source of illumination. And you can get a much greater level of resolution using an electron microscope versus a light microscope. These were first developed in the 1930s. But to see something, it first of all has to be placed in a vacuum where there's no air. Often the specimens need to be coated in a reflective material like a metal. So typically you don't see living specimens in an electron microscope. But they can give really high levels of magnification, up to 10 million times. So they'd often be used to observe cell organelles or bacteria or viruses, very small things. And the reason they can give such high resolution is because they have a much smaller wavelength compared to visible light. This is one of the first electron microscopes. Okay? You have a source of electrons, you have a vacuum tube, okay? You get a viewing piece here. A modern electron microscope basically just looks like a computer. This is the electron microscope part. Your specimen goes in here. The electron source is here. Okay? And you don't look through a microscope to see it, what's inside, you look at a computer screen. They have one over at Whitesboro High School. They got a grant to purchase one. It's kind of neat. I got to use it. This is an image I took in the electron microscope of a feather from an owl. There's a couple different types of electron microscopes. There's the transmission electron microscope, the TEM. This is kind of like the compound microscope. Light passes through the specimen to make the image. This is the one that gives the greatest magnification. The electrons travel through a thin slice of the specimen, and then you can see it. It gives you sort of a two-dimensional view. A scanning electron microscope works in a similar way to our dissecting microscope. Rather than electrons going through the specimen, they bounce off of it and are reflected, and that makes the image. So this is what gives a three-dimensional appearance, because the electrons are bouncing off the surface. This is a mitochondria viewed through a transmission electron microscope.
Now, how is that different in a mitochondria and any other microscope?
Well, just like if you were using a light microscope, the mitochondria are tiny, you could hardly see them. It's mostly about the level of resolution and magnification. Yeah. And here is a scanning electron microscope. Here's an ant. Probably it had to be covered in a metal, put into the vacuum tube, and then electrons are bounced off of it, creating this three-dimensional view. Sometimes you'll see scanning electron microscope images that are color. Those are artificially colored. The electron microscope gives a black and white image, so sometimes people color them to make different parts stand out. There's a few other types of microscopes. You don't really need to know the details, but I'll just mention them. You may use some of them in college at some point. Scanning, interferometric, apertureless microscopes use light, but they produce a much better resolution than a typical light microscope. The confocal laser scanning microscope uses a laser beam of visible light to create the image. Again, giving a good high level of detail. Phase contrast microscopes are popular at higher levels. They use differences in the refraction to sort of make cell parts stand out from each other. Okay, this is this confocal microscope. You can see the different parts of the cell more clearly. This is a phase contrast microscope. A few other biological tools. Sometimes biologists have to do very fine manipulation of microscopic things. For example, to do cloning, to take the nucleus out of a cell, they have to use a microscope, puncture a cell with a tiny little tool, use a little micropipette to suck out the nucleus, and they can insert a new nucleus into it. But this uses these tiny microscopic tools, and so they can perform very fine procedures. It's actually almost like you're doing surgery on an individual cell. A centrifuge is a tool. I have one up here. This is a centrifuge. Basically, you could put a test tube with a sample, a liquid sample, in these sleeves. Turn it on. Spins super fast. This one's pretty old. I don't know if it's really usable anymore. Well, when it's spinning so fast, what happens is the parts of the liquid separate by density. The most dense parts sink to the bottom. The least dense move to the top. So you separate the parts of a liquid by spinning them very quickly. And then you could take out the various parts and analyze them, work with them, etc. An ultracentrifuge spins even faster and separates things to an even greater degree. Tissue cultures are a really important tool biologists use. A tissue culture is when you take samples of a tissue from a living organism and grow it in a petri dish, okay, using some sort of culture, a medium that will keep it alive. And so this is important. For example, one way tissue culture is used is, let's say a researcher studying, say, breast cancer. They can take a sample of cells from a person that has breast cancer and culture them in a petri dish and have growing in their lab cancerous cells from the specific type of breast cancer. Then they can use they can try to treat those cells using different medications, different techniques, and see if it's working at controlling the growth of those cells in the lab, rather than doing that in an actual person. And so there are cell culture lines you can buy. You can buy all sorts of cell tissue cultures for a variety of diseases and tissues that you can then do your research using. So tissue culture is it's a growing field. One interesting question or topic that's coming up is now scientists are able to, I don't know if you call them scientists, but people are able to take a culture of animal tissue, muscle tissue, and grow pieces of meat in a lab rather than in an animal. So you could take stem cells from a cow and you can use them to grow basically a hamburger in a factory somewhere. And so one of the questions people say is, well, is that, so if I was a vegetarian, would I be willing to eat tissue cultured meat? It's still animal tissue, it's the same muscle fibers, same cells that make up regular meat, but a cow didn't grow and live and die to produce that meat. So does that solve some people's ethical questions about eating meat?
Would you eat that meat, Mr. Duras?
I don't think I would.
Yeah.
I mean, I don't have any desire really to eat meat, so that's probably part of the
Is it more that you
reason.
just don't like meat? than-
Yeah. So I don't know. But it could be a way to raise, so there are huge environmental costs that come about with raising animals for meat. And so creating meat products, even though it's real tissue, but creating them in this other way may be a more environmentally friendly way of creating meat. That's the question. This is what a micromanipulator, so you can see on the screen, there's a cell here in the middle, and this scientist is probably, they're inserting something into the cell using this tiny little needle. She controls it using the tool here, and then looks at it under the microscope there. This is another version of a centrifuge. And this is a blood sample, the color's off, but this is a blood sample after being centrifuged with white blood cells, plasma, and red blood cells separated out. And tissue culture, just a way of raising cells outside of a living organism. Last couple slides here. So often we will want to separate small parts of a solution. We have a few ways to do that. One is chromatography. This is using a special paper to allow different portions of a solution to be separated. We'll do this at some point this year. Basically, you take a sample of the solution, you put it on a piece of paper. You then hang that paper so that the bottom is just barely in some solvent. And as the solvent gets soaked off the paper, the different parts of the solution separate from each other, forming different bands. Electrophoresis, this is an electrophoresis chamber. We'll use this when we get to our genetics unit. So this electrophoresis is a way of separating DNA fragments from each other, or maybe amino acid fragments. If you've ever heard of like DNA profiling or a DNA fingerprint, okay, that they're created sometimes using electrophoresis. Spectrometry is a way of analyzing which elements make up a certain unknown material. A mass spectrometer is used. You can have an unknown sample, and the readings come back tell you, well, it has 64% nitrogen, 22% phosphorus, and give you a reading about what it's made of. And that's because different elements absorb light differently. This is what chromatography looks like. If you were to take a marker and put a dot on 4 pieces of chromatography paper and soak them in a solvent, the parts would separate out from each other, the different pigments that make up that color. Here's another. These are plant extracts, which all the pigments and leaves have separated out through chromatography. This is electrophoresis chamber in use. And these are the results of electrophoresis. And that's a mass spectrometer where you put a sample in to tell you what it's made out of. Then finally, our last slide, imaging. Sometimes, especially medical research, images of organisms, especially humans, need to be made. CAT scan you probably have heard of. Uses x-rays to produce an image. In soft tissue and bone, and different densities, absorb x-rays at different levels, and that can be used to create an image. Probably heard of an MRI. Maybe some of you had an MRI if you had some sort of injury or something. An MRI doesn't use radiation, it uses large, very, very powerful electromagnets. You sort of arrange the nuclei of atoms and then sense their orientation.
No way to take off the little metal stuff.
Yeah, it's really important. I have a good friend of mine is a radiologist, and he talks about sometimes accidents when, if you have an MRI, they tell you have to leave all your clothes, anything with any metal outside of the room, because if they turn on the magnet, there's anything in the room that can be attracted to it, it just goes flying at the magnet.
What if like metal meat?
Yeah, usually those are non-attractive to magnets, but you have to tell them that.
Isn't that why you don't need to go through the airport still?
What's that?
Isn't it, because it's like.
Sometimes, yeah. Sonograms use use sound waves to create an image. Okay, like in pregnancy. Or if you get a soft tissue injury, you might get a sonogram of it, an ultrasound. Yeah. Oh yeah, I need your toothpick lab. I didn't get it. Have a good day. See you tomorrow. See you later.